Welcome back from the coffee break. I hate to be a party pooper, but we do have some wonderful things coming up. If you would be lovely enough to start taking your seats again, that would be fantastic. I'm so happy we're having all these lively discussions already. This is really great. But we want to fill your heads with more ideas and more inspiration so you have cool things to talk about after the end of the day officially as well. So as everybody's entering here, we can slowly get started again. I think we're still waiting for a few people to slide on in from the coffee break. Okay, wonderful. And with that, an official welcome back from the coffee break. I know that the times are getting too short to discuss everything everybody wants to discuss, but we, unfortunately or fortunately, have more exciting things to tell you about. Our next keynote is going to be about something very exciting. We saw in the previous panel that there is a need for doers in academia, in research, in universities. And I think you're about to see what a doer does when it comes to innovation and technology and science. Professor Eran Segal is a professor of computational systems biology at the Weizmann Institute of Science. And his main goal at this week's conference is to tell you about the Human Phenotype project, which asks the question of what if we had decades of data about lifestyle, microbiome, absolutely everything that we could get from a human perspective. What could we do with it? Could we change health? Could we change diagnostics? Could we change the way we think about humans in general? Well, I definitely can't answer that question, but I'm really excited to see what Professor Eran Segal does. So please welcome Professor Eran Segal with the talk about cracking the phenotype.
Good? Yeah. Okay, hello everybody, and very exciting to be here. Thanks for the kind invitation, for the kind introduction, and the opportunity to tell you about the Human Phenotype Project, which we've been working on for the past several years. And the motivation for this project actually came from seeing what data brought to the field of medicine in general. And in my view, the most impressive project is the Human Genome Project, which, as you know, brought us the first draft of the human genome about 25 years ago. And if you look where this took us, it really was transformative. So before the Human Genome Project, we knew about several variants involved in just a few diseases. And 25 years later, we now know about hundreds and sometimes even thousands of genetic variants involved in virtually every single disease after we've genotyped tens of millions of people. And it's also been shown that if you use genetic evidence when developing drugs, that improves the probability of success, so reduces the cost of drug development by 50%. It's still in the billions, but that's a big advance. But of course, the genome is just what we're born with, and it doesn't take into account anything that happens to us throughout life, our nutrition, exercise, dietary habits, and so on and so forth. And that really what motivated us to start a few years ago what we call the Human Phenotype Project, where the idea is that in addition to the human genome, we want to measure everything that we can about people. So what we did was set up a clinic at the Weizmann Institute and really went very broad, very deep in terms of what we profile about people. So before even people come in, they upload online their electronic health records, so we know all the medications they're taking, any indications they have, all their historical blood tests. They fill out hundreds of questionnaires about medical background, family history of disease. Then they come on site, and they do a bunch of clinical and physiological and multi-omic measurements. So these include various body measures, blood tests to standardize across people, hand grip strength, gait analysis, ECG. We record voice that has correlates to disease. We take a bunch of imaging modalities, so DEXA for full body scan and bone density, high-resolution images of the retina, ultrasound to look at fat in the liver, and the carotids for cardiovascular health. People go home with sensors, smartwatches to track physical activity, sleeping devices that for 3 nights track various analog signals throughout 3 different nights, at the grade of a sleeping lab, except that people do it from the comfort of their home. Continuous glucose monitors, CGMs, that track glucose levels in the blood every 15 minutes for the course of 2 weeks. During those 2 weeks, people log their diet, all the medication they take, any physical activity, and we biobank samples. We have tens of thousands of aliquots from live cells, PBMCs, that we can do functional assays on, as well as serum. And we connect all of this to state-of-the-art multi-omic analysis. So we do whole genetic sequencing, but also vaginal, oral, and gut microbiome metabolomics that we do both targeted and untargeted. Metabolomics looks at thousands of different small molecules in the blood. Proteomics looks at proteins in the blood, RNA sequencing at levels of RNA, and we have a novel immune assay that I'll show later, whereby we can tell the entire infectious history of a person in just one experiment. So this profile is what each participant undergoes. It's a very deep, as far as I know, the most deeply characterized cohort that exists. We profiled so far 13,000 people in Israel. It's longitudinal, so every 2 years people come back to the clinic and undergo all of these different tests. Every 1 year, virtually, we call them to see any update in their medical condition. This is just an example of one of these many unstructured data sets that we have. This is the gait analysis, where we video people either walking or doing this sit-to-stand test. And from these, we can then extract the skeleton of people at 40 different joints and 60 frames per second, so really recapitulate the entire motion of a person. And we know that motion, movement, has many correlates to different diseases. If we look at all this data, it covers around 17 different systems of the human body, so very comprehensive. People that we enroll in the study are coming from the general population, so we have many different, 40 different diseases represented in this cohort, 30 different medical conditions, not quite diseases, but still interesting to study, like allergies, vitamin deficiencies, and so on. And we're now in the process of significantly expanding this cohort, where we want to grow it to a much larger number. So the next milestone is to hit 100,000, which we hope to achieve both by increasing the cohort in Israel, but also by opening additional sites in other countries. And actually, the second site that very excitingly we just opened a couple months ago is here in Japan, with the great help of our friends here. And we have a clinic that we're going to be opening up in Abu Dhabi in the coming months, and hopefully in additional countries very soon. And this really allows us to be also the first cohort that will be international, and to really tap into genetic, ethnic, cultural, and many other types of diversity from which we can discover many novel discoveries. Of course, we are not the only one to track human cohorts. There's multiple such projects worldwide. Typically, these are, by the way, run not by a lab, but by countries. The most prominent example is the UK Biobank, 500,000 people about 18 years ago that have been tracked longitudinally. Very impressive project with enormous impact. It really changed the way we do biomedical research. If you look at the number of publications, it took quite a few years, but once this ramped up, it's really growing every year. Over 10,000 publications by 40,000 researchers from 100 countries. Major investments by pharma companies who are paying to add additional data in return for exclusivity for the data for about 6 to 9 months, and then the data goes into the public domain. So in comparison, of course, the Human Phenotype Project, or HPP for short, is still smaller, but we have several of these unique features that are not part of the UK Biobank, like being international, as I mentioned, and then all these other state-of-the-art methodologies that did not exist when UK Biobank started, which include various of these multi-omic measurements like microbiome, RNA sequencing, immune profiling, and untargeted metabolomics, where we actually have the largest cohort worldwide, as well as some of these more deeper phenotypes, like characterizing sleep, Also, I believe at this level, the largest cohort in the world, also for CGM, liver ultrasound, and gait, and some of the other tests that we're doing. So by analogy to what happened and the impact that was created by the UK Biobank, I really hope that over the years, the HPP data set will also be able to create at least as much impact. And in order to do that, we are also now providing access to academics. There's dozens of actually world-leading universities and researchers who already gained access and are working on this data. But so far, it's been mostly discoveries from the research we've been doing in the lab. And this I'm showing you just to highlight the fact that there's already quite a lot of discoveries that are gaining a lot of interest. So by that, I hope that will be indeed creating impact over the years. And so in the remainder of the talk, I want to tell you about a few of these discoveries from the bird's eye view so that I can cover a bunch of these. So I won't go deep into any of these, but I'll just give you hopefully an overview of the breadths of types of discoveries and insights we can gain from such a comprehensive data set. So one of the earlier projects that we've been working on is linking the microbiome and nutrition to try and understand what is healthy nutrition for people. And the way we've done that is by looking at the CGM, continuous glucose monitoring of people, initially a group of 1,000 people. And one of the key discoveries was that if you consider glucose levels as a key indicator of health, which we know it is and important for obesity, diabetes, and many other diseases, then when we profiled different people, we saw that different people have very different blood glucose response to food, even when they eat exactly the same food. And you can see that here anecdotally for 4 participants, but we saw this on the scale of 1,000 people, where different colors represent different individuals. The same color represents the same individual eating the same 4 slices of bread on 2 different mornings. And you can see very reproducible blood glucose responses in the 2 hours after eating a meal within a person, but very different responses across people. And so this data showed us that if we want to control blood glucose levels across people, we have to personalize the diet in people because the same food elicits different responses. And indeed, by integrating clinical parameters as well as microbiome information, we could develop a machine learning algorithm that quite accurately predicted these blood glucose responses in people. So here, the axes correspond to the area under the glucose curve in the 2 hours eating a meal, either predicted by the model or measured by the CGM. And you can see fairly good correspondence such that we could take individuals who are pre-diabetic, who for 1 week on their habitual diet have many spikes in their glucose levels, and we could personalize a diet for them based on the algorithm that could fully balance their diet for the same individual just 1 week later, eating exactly the same amount of calories with surprising foods, including sometimes ice cream, pizza, and so on. Not all the time, but sometimes. So we then took this to the next level and did a fully blown randomized clinical trial on 200 people with prediabetes that we randomized either into the standard of care Mediterranean diet or the algorithm-based diet. Anecdotally, this is 1 participant in the 1 month prior to the intervention. You can see many spikes in glucose levels. And then the first month of the intervention on the algorithm diet, pretty much full normalization of blood glucose levels. And looking statistically, if you focus here on the center panel, this is the change in hemoglobin A1C, the gold standard marker for looking at diabetes. It measures the average glucose levels in the past 3 months. You can see on the Mediterranean diet a significant reduction, but double the effect on the algorithm diet and many other improvements in other metabolic parameters. So for us, this was very gratifying to go. It took us about 10 years to go, but to go from an idea to developing an algorithm to an actual intervention in a randomized clinical trial that we consider as a treatment for people with prediabetes. And we also showed this to be effective for people with type 2 diabetes. We have a major interest in the microbiome and its relations to health and disease. And so in another project, we dug much deeper into looking at the microbiome, and we took a novel approach whereby, in contrast to how we typically in the literature have been looking at the bacteria in terms of the composition of bacteria, we went into a higher resolution and looked at the genetic variation that we see within the same species of bacteria across different people. So what I mean by that is that this is just an illustration. If we have 3 people and we profile their gut bacteria, we can then map the sequencing reads that we get onto the genomes of different bacteria, and then record the variation that we see at every position of any given bacteria, and then try to associate that with some trait of the host, in this case with BMI. So in this illustration here, you can see that it's a little offset, but there's an A at this position for the first individual and a BMI of 31 compared to a lower BMI for the other 2 individuals who have a T at this position. And so this we would consider as some association between genetic variation at a particular position in one bacterial species and a host trait. This is, of course, an illustration, so we do the analysis on the data of thousands of individuals, and then we can plot it as what's called a Manhattan plot that is commonly used for looking at human genetic analysis, but now the x-axis corresponds to different species of bacteria. Every dot here corresponds to one of these positions in one particular bacteria, and the y-axis is the significance of the association, the minus log p-value. So you can see that we get many associations that are actually very significant, but even more interesting than the fact that we get many significant hits is actually the size of these effects. So if we now plot exactly the same data, every dot is still one of these positions in one bacteria, the y-axis is still the significance, but now the x-axis is the number of BMI points that are statistically explained by this variation at one position of a bacteria, and how many points of BMI it can explain. You can see that we actually get many hits that are with a significance of 1, sometimes even 2 points of BMI. That's a huge effect. It could be explaining about 10 kilograms of body weight just by a genetic change in one position in one bacterial species. In fact, it's bigger than the biggest association that was ever found in the human genome, which is around 1.3 points of BMI explained by the SNP in the human genome that has the biggest effect. And this is just to illustrate. So here we have about 400 individuals that in one particular bacteria have one of these, one type of mutation. And if we compare them to 2,000 individuals that have just at this particular one position in the genome a different DNA base, there's already a 2-point difference in BMI, which is a huge effect, and this group of 2,000 individuals have also the same the same level of BMI as do individuals that don't even have this bacteria. So this is an example of a major statistical effect on BMI that is explained by these bacteria. We have many more of these also for many other indications. Of course, the next step is to prove causality, whereby we would have to go isolate the relevant bacterial strain from these lean individuals, give them to individuals who are overweight, and see whether that actually has some benefits for the trait, in this case for obesity, in other cases for the other types of traits that we're looking at. But this is the type of framework and pipeline that this microbiome work can generate. I mentioned before that we have a novel profile of the immune system. So the way this is set up is we actually can synthesize, there's companies that can synthesize for us hundreds of thousands of different antigens. And then through a process of what's called a phage display, we can incubate all of these antigens together with antibodies that we have in the blood, in the serum of a person. And then by sequencing, we can tell all of the antigens of a person that are actually bound by that person's antibodies. What we did in this first library was actually to design 250,000 antigens that originate from gut bacteria and then incubate them to see which, for each individual, which of these antigens are actually recognized by that person. And what we found is actually that there's, in fact, many, many different of these antigens that are very specific, that are recognized by a very, very small number of people, sometimes by even just one individual from the cohort. But then other of these antigens are actually very broad. They're recognized by many individuals, sometimes approaching most individuals. And on average, there's about 1,000 different antigens recognized by a person. Moreover, this is actually a person-specific signature. So if we compare the same individual to himself 5 years apart, we see that different individuals have low correlation, but still some correlation because people were probably exposed to similar environments, similar bacteria. But the same individual actually has a very high correlation to himself, retaining this immune profile. And we can then take this, and this is just one example of multiple different disease associations that we found, to look at whether we find specific immune profile signatures for disease. So this is a small cohort that we looked at for chronic fatigue syndrome patients, and we actually identified multiple different antigens, in this case antigens originating from flagellants, which are the motors of bacteria, and we found them to be much more elevated in chronic fatigue syndrome patients, such that we could even develop a computational diagnosis tool whereby, based on the immune profiling, we can tell with pretty good accuracy which individuals suffer from chronic fatigue syndrome and which individuals do not. And this is a condition where very few, if any, biomarkers exist. So this is just to show you one example where this type of data can help shed light on. Another type of very informative data that we were profiling is the set of metabolites. So these are all the small molecules that we can profile in the blood of a person. And in one of these assays, we basically can separate with a machine that separates compounds based on their mass. We can identify thousands of different small molecules in the blood. So in previous work a few years ago, we asked a very fundamental question, which is what determines levels of all of these different small molecules? Because we know that small molecules are very key for our health, everything that circulates in our bloodstream. And so once you identify a molecule that you suspect is involved in some disease, the next thing that you'll want to do is you'll want to modulate, you want to change the levels. And in order to do that, you need to know what controls that molecule. So what we did is we took advantage of the HPP that actually has many different types of measurements per person, and we asked this very simple question of, for each metabolite, which of these different sources of data can explain it best? And what we found is that for some molecules, actually it was gut bacteria, and we could even identify specific bacteria within the microbiome that were best explaining levels of that metabolite. Whereas for other metabolites, it was dietary factors that determines its levels. A simple example is levels of caffeine. One of the small molecules that we measure is determined by, not surprisingly, how much coffee people drink. And then other small molecules were determined by various genetic factors. So this helped us to actually build a database where for each metabolite we could identify whether that metabolite is being controlled by microbiome, diet, genetics, lifestyle, exercise, or other different of these sources of data. And then using this data, we could then shed light in specific cases of various diseases, and I want to show you one such example. So here we looked at even just a single patient, acute coronary syndrome patient, so somebody who underwent a heart attack. And we can take this, let's say, 60-year-old male, and we can go into our large-scale healthy HPP cohort and build a specific personalized cohort for this 60-year-old male. We could basically identify 60-year-old males who are healthy within our cohort. And then we could look at this metabolomic data And for each metabolite, we could look at this reference group, the healthy cohort that is matched for our one patient, and we can build a distribution of what the levels of this metabolite should be in a normal patient population, the reference for this patient. And then we can see where the level of that metabolite is for that person. In this case, we can see that it falls actually outside of the distribution. It has a very low value. We can quantify that by the number of standard deviations away. We can do that then for all the different metabolites. And by that, we can obtain a metabolite deviation profile that is specific to this one patient. We can then cross it with the database I showed you before to ask the question of which of these, for the metabolite deviations, what is actually determining these deviations? Is it diet, microbiome, genetics, or other traditional risk factors? And when we do that, we find that even though we can have two 60-year-old males who present at the clinic with the same condition, they both underwent a heart attack, when we analyze their metabolite deviation profiles, we see that the underlying reason, the pathophysiology of disease may be different, where in one individual it could be driven by microbiome, in another by diet, in another by genetics, in another by a combination of these or by other factors. So this really might give us hints as to why the same type of treatment doesn't work for all people, and why and actually what personalized treatments we need to give people. Another area of great interest to us is longevity and wellness and biological aging. And this is an example of a project that actually leverages altogether all of the different sources of data that we have. So going again to just the metabolomics data, basically we took this data, again, thousands of different small molecules in the blood that we can measure about a person. And we developed a machine learning algorithm that, based only on the metabolomics data, tries to predict the actual chronological age of a person. So the predictions are in the y-axis, and you can see a pretty good correspondence. Every dot corresponds to one individual. But you can see that these predictions are also not perfect. So people here at the top, when at least the model looks at their metabolomic profile, they actually look to the model more like somebody who's with an older age. And conversely, people at the bottom to the model look like somebody who's actually younger. So we asked the question whether these errors of the model are actually really errors, or do they really capture some actual biology? So comparing the individuals at the top that, according to the model, have an accelerated age, they have a metabolomic profile that looks like somebody older, to individuals here at the bottom, we found that, in fact, these are not errors of the model, but individuals here that have a higher biological or metabolomic age, they're in worse clinical condition. They have higher BMI, higher triglycerides, hemoglobin A1C, blood pressure, cholesterol, and liver enzymes. So they really have a worse clinical condition that could be identified from the metabolomics data without integrating this additional clinical data. And so much like we can do this for the metabolomic data, we can do it for any of the other sources of data that we have. So this is the RNA expression data that we measure from the blood. Again, we can see fairly good predictions, but also these errors or individuals that have seem to have accelerated age, RNA age in this case, compared to lower age, and indeed people with a higher RNA age are also in worse clinical condition by several of these other measures. And we can do this for the other types of data. This is from the retina scans, the only place where we can really look at blood vessels non-invasively. And so the original image that we take is this one, and then the other images are what a deep learning algorithm actually sees underneath. And then we can extract also here features that change with age. And by and large, one surprising system that we found, one surprising finding that we found is that all of these different biological clocks, and we built 17 different biological clocks for 17 different systems of the body, by and large, they're independent. This is actually a result that surprised us. So it means that if you're aging faster in one system of the body, you don't necessarily age faster in another system of the body, where naively I would think that if you're deteriorating across multiple body systems. But even though there's some correlation between these clocks, by and large, they're independent. So these body system specific clocks not only give you some risk of disease, but they give you risk of a specific disease related to the body organ on which the clock is developed. And we show this in this plot here. For example, a biological clock that we have for insulin resistance, if you have a higher age there, you're at higher risk for developing a disease, not just any disease, but specifically diabetes related to insulin resistance, of course. And the final result that we had was a marked difference that we saw between males and females, whereby males we see age pretty much linearly, whereas in females we see this big jump between the age of 50 and 55, which we could actually link to menopause, because if we compared women of the same chronological age, post versus pre-menopausal women, we saw that post-menopausal women actually have a higher biological age for multiple of these different clocks as compared to premenopausal women of the same chronological age. And finally, these clocks actually were also, we also showed they had prognostic value. So if we start at some point, and now we leverage the longitudinal data, so the fact that we track people over time, we can see that people who have a higher biological age to begin with, they're at higher risk of developing a disease. In this case, these are cardiovascular incidents of disease as compared to individuals of the same chronological age, but different, but actually a younger biological age. And we see this also for new diabetes indications. And so these clocks also have prognostic value into development of disease in the future. And so now in the final few minutes of the talk, I want to shift gears a bit and tell you about the newer types of methodologies and models we've been developing this and kind of the longer term vision, which we actually are beginning to realize right now, which is to take advantage of the next generation AI models, the types of models that power applications like ChatGPT that underlie large language models, and bring them to the medical domain, to all the HPP data that we're collecting. And so the vision is that because we measure many, many different types of data, about 30 or 40 different modalities on a person, and each of them gives us a different angle, a different view into human health, the idea is that by using these types of AI models, we want to align all of these different types of modalities and get a more holistic view, a more holistic model of human health. And so on the way to building that, we started by building these types of AI models for individual modalities, and then so for some pairs of modalities, eventually building up into putting all of them together, which I will also show in the end. So one of the more successful models that we were able to build off of this is a model where we took our CGM data, the glucose measurements that we have for each person, and we tied them together with this new generation of AI models. Often the architecture is called transformer models, and so we call this a gluformer model, a model that is about the CGM, the continuous glucose monitoring data. And the idea is that much like large language models are trained to predict the next word in a sentence, these models, in our case, are now trained to predict the next glucose value of a person. So what that means is that once we actually observe some history of a person, some glucose profile of a person from the beginning of the day or from the past few days, we can generate, these are generative AI models, we can generate plausible completions of what the CGM of that person would look like for the rest of the day and even for future days. But much more interesting than that is that the representations that we then learn about a person, we found are very useful for predicting future onset of disease. Much like in large language models, it's not just about predicting the next word, but it's just that the representations allow you to capture something very fundamental about language that then allows you to do various applications like translation, chat, and many others. So here's what I mean by that. So here we took a group of people who are pre-diabetic. What that means is that their hemoglobin A1C is between some range, 5.7 to 6.4. And we saw that, quite surprisingly, if you look at their levels of hemoglobin A1C, then individuals who are at the higher range are actually, over time, if you look 2 years into the future, not more likely to develop diabetes than are individuals who are at the lower range of hemoglobin A1C, which actually is a surprising result, but it shows you that hemoglobin A1C by itself, even though it's the gold standard, is not useful within the population of pre-diabetics to tell you who will develop diabetes and who will not. So in other words, who you should treat and who you should not, because they're not going to develop diabetes. But in contrast, when we took the gluformer model and we asked it to predict which individuals would develop diabetes in the future, here we saw a very clear separation, and even across a cohort that we never had access to when developing the model, another cohort from Spain that actually had 10 years of longitudinal data, we see again that hemoglobin A1C itself doesn't tell you which individuals will develop diabetes in the future and who will not, whereas the gluformer model was able to very significantly separate those individuals who are pre-diabetic, but very few of them 10 years down the road will not develop diabetes, from those who are much more likely, actually 25% likely, to develop diabetes in the future. So this model, we believe, now has great utility for the pre-diabetic population, for example, and for many others, to identify individuals that should receive intervention versus those who are pre-diabetic by their levels of hemoglobin A1C, but perhaps this is due to other factors, genetic factors or other factors, but they are unlikely to develop diabetes. So as I mentioned, we now are developing these models for other types of modalities. We recently developed one for the microbiome, which we call MBformer. You'll see this pattern repeat itself for different modalities. Here, the interesting finding is that when we link it to various traits, like, for example, age, linking microbiome to age, we see that actually the model, the foundation model, does a much better job at generalizing across different populations, different cohorts, than do models that are just trained traditionally on the microbiome abundance data. Another model that we developed is one for ECGs. This one I'm showing you because it's a different type of learning algorithm. So here, instead of trying to predict the next word like we do with glucose levels, here we take this approach of contrastive learning. So this is trying to align different types of data. So here we basically take the first 5 seconds and the last 5 seconds of a person's ECG, and we ask the model to basically pull together the embedding, the representation that we learn for these ECGs if they come from the same person, and push them apart if they're coming from different people, or pull them together if they're coming from the same person across different visits 2 years apart, and push them away if they're coming from different people. And much like you can align text with images, we find that this allows us to actually align the ECG of a person with himself, and also have utility in predicting various biomarkers. And here's another model that where we tied a pair of 2 different modalities, the retina scans and the carotid ultrasound images, because both of them are informative of cardiovascular health, except that they give us information from 2 different angles. And if you ask the doctor, given the retina scan of a person, to identify within a database now of 10,000 carotid ultrasound images, which carotid ultrasound images corresponds to the patient for which you've given him the retina scan, a doctor can't do this task, but a learning algorithm that, again, by this contrastive learning approach, actually can do a fairly good job at linking together the 2 different imaging, indicating that the model actually captured something fundamental about cardiovascular health in this case. And so this is a subset of the types of individual models that we're building for a single modality or sometimes for a pair of modalities that already have usefulness, but really the vision and the model that we're already building, and I think within the next few months we'll have the first version with already some promising preliminary results, is a model that we call HealthFormer, where here the idea really is to tie in all of the different modalities that we measure about a person. So if you imagine all the 30 or 40 different modalities that we have about a person, one approach by which we can do this that we started implementing is to put all of the data that we have on a person on a timeline, much like we do with the glucose value, except that now we have many different types of data. So it's not just glucose levels, but we have all of the different medical events that we measure about a person. It could be the next blood test we measure about a person, the next indication of disease, the next medication a person will take, and so on and so forth. So we can put all of these on a timeline, and then we can do the same type of model whereby instead of just predicting the next glucose value, now we predict the next medical event of a person. When we do that, if you look at all of the different modalities that we have, and of course they have various different sizes of data, because some of these are actually measured several times per second, whereas some of these, like blood tests, are measured maybe once every year or two. But if you look at all of this data together, now we have about 20 million data points per person, and if we have about 20,000 visits per person, that's already hundreds of billions of data points that allows us to learn these types of models. And what I think is really interesting is that if we can build these models, then these models could really act as a digital twin of a person, a personalized digital twin of a person. Personalized because if you feed it the history of one person, the health trajectory predictions that you'll get will be different for one person versus the other because they have different histories. But what you can then do is you can actually then simulate within your digital twin different interventions. So you could say, what would happen if I ate more bananas, less bread, I swam more, I drank more alcohol, and so on? What would be the effect that that would have on my cholesterol levels, and so on and so forth, for different interventions on different types of measures and indications. And so this is really the vision that we're building towards, and the idea is that as you collect more data per person and more different types of modalities, and as you collect more data from more people, the accuracy of these digital twins will improve over time, allowing you to really build and have, hopefully one day in your app, a digital twin of yourself, whereby you can really simulate future health trajectories, including simulating the effect of different interventions. And this app would be updated and update itself as you collect more and more information about yourself. So in order to do that, as I mentioned, we're now trying to significantly expand the size of the HPP cohort to grow by more people, more different phenotypes, more diseases, and also more countries, so that we can look at additional diversity and learn from that. And that's why it's really very exciting that we finally were able to launch the clinic here in Japan, and hopefully soon in other countries. And I really think that it's this type of data sets that will really allow us to build the next generation of foundation AI models for precision medicine. And I'll stop here. Thank you very much.
Thank you so much for presenting this really inspiring vision for the future of health. In lieu of a Q&A segment, we actually have a treat for you. In about 10 minutes after you go on a little coffee break and return, we have a really exciting panel where we get to see where the future of precision health, backed by this kind of research, is really leading. We have scientists and entrepreneurs that are going to take the knowledge you have gained in this keynote session and give us the context that we need going into the future. So let's thank our keynote speaker once again and take a little short coffee break. And we are returning here with the panel at 4 p.m. Thank you so much.